a psychiatrist, and I'm the Executive Director of the Mindgardens Neuroscience Network. That's a network that's based here on the Randwick Health and Innovation Precinct, and it's a partnership between the University of New South Wales, South Eastern Sydney Local Health District, Black Dog Institute, and Neuroscience Research Australia, and it collaborates with our other partners as well. The important part about Mindgardens and the precinct is the bringing together of different groups of people, both from a clinical, service management, consumer and research lens, to actually optimise people's wellbeing and outcomes. So for Mindgardens, we focus on neuroscience, mental health and addictions. In particular, we run a range of capacity building programs and projects and workshops for clinicians. We have very consumer focused co-design workshops and a whole lot of other translational research activities. If you'd like to find out more about Mindgardens, reach out to our website. There's contact details there, and please feel free to come along and be part of that broader network of people thinking about neuroscience, mental health and addictions. I'm just commenting that this program today is being recorded and will be available later for everybody to see. So it's my great pleasure today to introduce two speakers from different ends of the spectrum in terms of the age ranges that they're focusing on this important topic of dementia. And I think this fact that the series today of the precinct bringing together different perspectives is really important, and something that we can learn from areas outside our own domain. So we're going to hear initially, and I'll introduce each speaker as they're coming up, and then there'll be time for questions after each speaker as well. But it really gives me great pleasure today to introduce Scientio Professor Kaarin Anstey, who's also an ARC Laureate Fellow in the School of Psychology. Professor Anstey is the Director of the Aging Futures Institute at UNSW and Senior Principal Research Scientist at Neura, one of our Mindgardens partners. So welcome, Kaarin, and over to you to talk about late life dementia. Thank you.
Thank you. So I would also like to acknowledge the traditional owners of the land, the Bedigal people here, and pay my respects to elders past and present, and to extend that respect to any Aboriginal or Torres Strait Islander people who are online with us today or here in the audience. And I'm talking about late life dementia. I'll just pick out a few topics within that, because it's obviously an enormous topic and I only have a short period of time. Not sure how this is.
Oh, just use the arrows.
The arrows aren't working, sorry.
Oh, hang on. Yeah, okay, so it's going to be a click.
Thank you. So I'm just going to give you a really bit of a cook's tour. First, talk about prevalence, some Australian data, some of the emerging topics in dementia, then just look more at my area of interest, which is in prevention. So how much of dementia is modifiable and what's some of the current research that we're doing. So global prevalence, our team conducted a synthesis of the global literature on the prevalence and incidence of dementia. It was published this year. We looked at nearly 1 million people in over 200 studies. And the prevalence of dementia increases with age, and it's highly prevalent in the 80s and 90s. It looks like it's more prevalent in women than men, but actually that's only statistically significant in the 90s. When we break it down by subtype, Alzheimer's disease is more prevalent in women than in men. However, in Australia, we don't have good prevalence data. We don't have a national study of dementia prevalence, and all of our estimates are really guesstimates or sort of intelligence guesstimates based on either international data or other types of data. And there's quite a variation. So Dementia Australia uses the NATSEM report, and it estimates that there's 487,000 people living with dementia over the age of 65. So I'm not talking about childhood dementia here, of course, or early onset dementia, younger onset dementia. The AIHW, on the other hand, has a lower estimate. They say there's about 400,000 people living with dementia. And then we analysed the census data and some other GP databases and published in the MJA this year, and we found even lower numbers of dementia in Australia. So it's really an open question at the moment as to how many people in Australia have dementia, and that's really critical in terms of future planning of health services. So it's actually was a priority that was identified by the Australian Institute of Health and Welfare in their data gaps report that was published in September. In addition to dementia, we have mild cognitive impairment, which is really a preclinical stage. And I like to combine mild cognitive impairment with dementia from an epidemiological perspective, because it really shows you the true burden of disease that we're looking at and just how prevalent this is. So if you look at, the over 70s, we've got 30% between 75 to 79 with either MCI or dementia, and over 90, it's over 60%. So when we combine these two categories, it's huge. And there is variation, though, according to race and ethnicity and a whole lot of other socio-demographic factors. Then, of course, we have social determinants of health. And we know that in the epidemiological work conducted actually out of Neuro by Kylie Radford and colleagues, dementia prevalence is 3 times higher in Indigenous Australians compared to the general population aged 60 and older. And there's a disparity there between urban and rural Indigenous Australians as well. So when we talk about dementia, that's an umbrella term for a number of causes of dementia. The most common cause of dementia in Australia is Alzheimer's disease. It accounts for about 65 to 70%. But when we actually look at autopsy, this is quite old data, but it's still relevant. When we look at autopsy, it's very rare to see Alzheimer's disease pathology alone or in isolation in late, dementia of old age. So usually people have mixed pathology. And I think that's quite important when we're starting to look at things like the new monoclonal antibodies to treat Alzheimer's disease, when we may end up treating that, but it's really vascular cognitive impairment that's causing the symptoms in a person. So understanding the underlying disease processes is important. So I just wanted to talk about some of the new developments in dementia, which are really important, and then I'll move on to prevention, which is really where most of my research is. I think this is one of the biggest developments that's very exciting, and it's going to really enable progress, is the plasma biomarkers for Alzheimer's disease that are now available using blood tests, and they're actually being used in clinical practice in some clinics in the US, in Europe. They're not yet being used in clinical practice in Australia. A number of pharmaceutical companies are going to bring out blood tests. They're going to become more and more common, and ultimately we will be seeing them used in primary care. GPs will be able to order these tests, and this is really going to completely transform the whole way we conceive of Alzheimer's disease. So there's a few of the biomarkers that are important. There's a plasma AB42/40 ratio, and that can detect abnormal amyloid status in either someone with cognitive impairment, but also in someone who's completely healthy. You can have an abnormal amyloid status, and that shows that you have Alzheimer's disease, but you're not yet symptomatic. There's also p-tau, and that's a very important marker. p-tau217 correlates with tau tangles in Alzheimer's disease. Specifically, it's elevated in the presence of amyloid plaques. In someone who is symptomatic Alzheimer's disease, it's increased by 300 to 700%, and it can detect AD pathology in adults 20 years before changes in cognitive status. And then we have the more generalized markers, the serum neurofilament light chain and the GFAP. So the accuracy of these tests is improving all the time. There are some limitations because they've been developed in research settings, and they're confounded by things like race, gender, and ethnicity. And the cutoffs, the clinical cutoffs in some of the US data do differ according to race and ethnicity. So it's really important that we get local Australian data on these, but we also have prospective studies collecting data and evaluating these longitudinally. So I think that's just that is on the horizon, and we will need to know about it. And I was at a recent workshop for training of GPs run by Health Ed, and there actually was a session on blood biomarkers at that. So this is coming online. And then alongside this, and really, in partnership with this, we have the new drugs for Alzheimer's disease, the anti-amyloid antibody treatments some of which have been shown to have clinical benefit. So there have been some anti-tau drugs. There was one that was just published this week, which was non-significant, but there are many other drugs in the pipeline. There's now 2 approved by the FDA and which are used in clinical practice in the US, that's aducanumab and lecanemab. The one that was announced at the US Alzheimer's meeting in July is donanemab. That seems to be the most effective drug. When you actually look at the results, which I'll show in the next slide, they're quite small. Now, these drugs there's been a lot of debate around them, and there's some people who are quite critical. They have quite significant side effects. They have what's called amyloid-related imaging abnormalities, ARIA. They're very common, especially in people who are ApoE4 positive. ApoE4 is the main genetic risk factor for Alzheimer's disease. So there's safety worries. There's also issues about how long should people take them for. And then if you look at the actual benefit, this is the data from the Trailblazer trial that was just published in JAMA. It's really, when you break it down, it's really only giving people a few months. It's delaying their Alzheimer's disease progression by a few months clinically. Interesting, if you look at the lower part of the slide, that shows you the amyloid burden. And there is almost a complete clearance of amyloid from the brain for patients taking these drugs, which is really interesting, but it's not accompanied by reversal of symptoms. So there's a whole lot of questions about, well, when should these drugs be started, et cetera, et cetera. But on the other hand, this is like the tip of the iceberg. There's lots of drugs in the pipeline. So we're moving to a really exciting phase with Alzheimer's disease. I just want to step back now and move towards prevention. So I want to talk about genetics. There is a well-known established genetic risk factor for Alzheimer's disease, the apolipoprotein 4. Allele1 copy increases the risk of Alzheimer's by 3-fold, and 2 copies increases the risk by 5 to 8-fold. And there are some other genetic contributors. There's genetic risk scores which have been developed. There's about 12 genes that increase the risk of Alzheimer's disease, but they have a minor effect compared to that. The other types of dementia, the genetic contribution is actually smaller in the late life. We're talking about the older dementias, the late life dementias, which is somewhat surprising sometimes. So when we're looking at risk reduction and prevention, we divide the risk factors into non-modifiable and modifiable. Obviously, genetic is a non-modifiable. In the modifiable, we tend to cluster them according to psychosocial, biomarker, physical health, and lifestyle factors. And then the question is, well, how much, when we're looking at dementia, how much is modifiable? You know, how important are genetics? How important are modifiable risk factors? If I have the genetic risk factors, should I bother with risk modification? Is it inevitable, etc.? Some of these really big data synthesis projects have tried to estimate the proportion of dementia that's attributable to modifiable risk factors. And this was the Lancet Commission that was published in 2020. And they estimate that globally, this is obviously making a huge lot of assumptions put across all different countries, assuming that, you know, risk factors are the same for everybody, etc. But globally, 40% of cases of dementia are attributable to these 12 risk factors, which includes psychosocial, medical, and even air pollution, so even environmental risk factors. We've conducted this analysis for Australia, and we actually find a higher population attributable risk in Australia. And the 2 biggest risk factors in Australia that account for dementia are insufficient physical activity and midlife obesity. So the question of how much dementia is attributable to genetics versus lifestyle was addressed in this big systematic review, and that looked at over 1 million people and tried to evaluate the contribution of lifestyle versus genetics. And what they found, so they made their reference group in the systematic review, the people that had low genetic risk, so they didn't have an ApoE4 allele, and they had a favourable lifestyle. So that's your reference group. That's your ideal. That's what we'd all like to be. Just by way of information, 1 in 4 of us is heterozygous for ApoE4. 1 in 4, so it's highly prevalent. So you have their reference group, and then if you look at the lifestyle, if you have an unfavourable lifestyle but no genetic risk, your hazard ratio is 1.52, so 50% increased risk just because of your unfavourable lifestyle. If you have a high genetic risk and an unfavourable lifestyle, your hazard ratio is 2.83, so very high. But there's still quite a benefit if you have the high genetic risk and a favourable lifestyle, it's much lower. So there's still a benefit in lifestyle modification regardless of your genetic risk. And the other important finding from this study and from others, including one from my lab, is that they're independent effects. So they're additive, the genetics and lifestyle effects in terms of their risk of dementia. So we tend to take a life course approach when we're thinking about dementia risk and dementia risk reduction. So there's some risk factors that are important that occur or are modified in early life. So less education in early life is a risk factor for dementia in late life, which indicates that we need to invest in early childhood education and retaining children in school. And globally, that accounts for the most dementia. In middle age, there's another cluster of risk factors. And then in late life, in middle age, it's really the vascular risk factors that are most important. So hypertension, diabetes, and obesity, midlife obesity. And then in late life, it's depression, social isolation, and some of the others, air pollution. So the vascular risk factors like hypertension and obesity are no longer risk factors when they occur in late life, which is quite interesting. So we need to take a life course approach when we're thinking about designing any type of preventive or risk reduction program. So I'm just going to name the risk factors because I don't have time to go through all the evidence. So the medical risk factors that are really well established are diabetes, heart disease, depression, stroke, kidney disease, hypertension in midlife, obesity in midlife, and head injury. This is just an example of a systematic review. So there's hundreds of systematic reviews. So people with type 2 diabetes are 49% more likely to develop mild cognitive impairment and 43% more likely to develop all-cause dementia or Alzheimer's disease. And this is an incredibly robust finding. It's been replicated many times. But it only accounts for about, I think it's like something like 4 to 6% of population attributable risk of dementia. So it's, in the bigger picture, it's not as important as some of the other modifiable risk factors. Then heart disease, individuals with coronary heart disease have on average a 45% increased risk of cognitive impairment or dementia. Blood pressure is probably one of the most important medical risk factor because it's something that GPs manage, on a day-to-day basis. And we know that blood pressure lowering reduces the risk of cognitive decline. And it doesn't matter which type of antihypertensive. There's been systematic reviews to try to evaluate whether there was a particular type of antihypertensive that's more beneficial, and none of them have shown any difference. So that's a really important one. And then obesity, which I've just mentioned. So the lifestyle risk factors are diet, physical activity, smoking, alcohol consumption, and poor sleep. The risk factors combine. So the more risk factors you have, the greater the risk of dementias. And that's now also been shown in individual studies and in systematic reviews. And because of that, researchers in dementia risk reduction really advocate for taking what we call a multi-domain risk reduction approach. So interventions that we're conducting address multiple risk factors in the one intervention. So we don't just do a dietary intervention or just do physical activity. We would advocate that it's better to do an intervention where you're addressing physical activity, diet, cognitive engagement. I haven't actually talked about low cognitive engagement. That's another lifestyle risk factor. So just in order to implement risk reduction and population health approaches, you need to be able to assess the risk factors for dementia. So we've spent a lot of work, a lot of time developing risk assessments. And this is a recent evidence-based risk assessment tool for dementia where you can go online and you can assess your risk. It's based on the Neuro website. This has been implemented and it's now, GPs are now being trained to use this through Health Ed, and it's being used as a professional outcome exercise to use this on 5 patients. And we've had quite a very good uptake with thousands of GPs and patients and people from the public using CogD risk because it's completely evidence-based and it gives a lot of information. Just by way of information, here shows you the relative size of the risk factors when you look at their relative risk, and you can see that depression actually has the largest effect in our meta-analysis. So it sort of shows you the importance of mental health for late life dementia. I just want to give you an example of one of our trials on dementia risk reduction that's underway. So we have a trial underway called MyCoach. It's a randomized controlled trial aiming to reduce risk in people with cognitive impairment or subjective cognitive decline. It's completely digitally delivered. So we developed this during COVID. So we do the entire intervention online, but it's done with people. When I say it's done online, it's not the neuropsych assessment, the baseline assessment and all the onboarding is done through a Zoom call. So you're interacting with a psychologist. We have a lot of phone support. And then we have a physical activity intervention with an exercise physiologist that's done remotely, but it's done online with a real person, and the same with our dietary intervention. And the intervention goes for 12 weeks, and we then have a 6-month and a 12-month follow-up. And our primary outcome is risk reduction, but we're also looking at cognitive impairment, quality of life, daily functioning, and psychological well-being. We have randomised, we've completed recruitment, and we've actually completed the intervention. So we have 326 participants who are currently in the trial, and we're now in the follow-up stage. And we have found that through our recruitment, we have 1/3 of our participants from regional and rural Australia. It's a national study, and 1/3 of our participants there. The economic benefits of prevention have been demonstrated through a cost-effective analysis of individual trials as well as simulation studies. And they've been clearly shown to be beneficial. That's really because dementia care is so expensive relative to an intervention. Even delaying dementia by 1 year is very beneficial. And if you delay it by 5 years, the cost benefit is enormous. And so, to summary and conclusion, late life dementia is highly prevalent, especially if you add in mild cognitive impairment. The new developments we need to be aware of and following are the plasma biomarkers and the modestly effective anti-amyloid treatments, but there's many more in the pipeline. Both genetic and lifestyle factors influence risk independently. There's a large scope to modify trajectories of cognitive decline in incident dementia through risk reduction. The effects of risk factors appear to be additive, indicating the value of addressing multiple risk factors. And risk reduction for promoting brain health is cost effective and has many benefits for healthy ageing, both individual and population level. Thank you.
Great, thank you so much, Kaarin. Stay up here because we'll see if there's any questions either in our live audience or online. Please. Yes.
Really lovely data.
Getting the microphone. So.
And I suppose the question I want to ask is, in the Danish economy is taken off because of their anti-diabetes drug, weight loss. And so the question I was really going to ask is, I think, you know, the importance of modifying lifestyle risk factors is really important. But what I worry about is people will see that there are drugs to treat Alzheimer's disease. And do you think there's going to be an issue where it'll be harder to actually get people to change behaviour if they believe that there's a drug or something that they could take that might either be preventative or treat? Because I think this is obviously a big population problem, and actually trying to get at the risk factors early is also really, really important.
That's a fantastic question. Thank you. I think it is a risk, but if they actually knew what was involved in taking those drugs, it wouldn't be very motivating. They have to have an MRI scan every 6 months. They have to have an infusion every 2 weeks. And there's 25% of participants have brain bleeds who take them. So, but I guess once the drugs become more effective, it is something we'll have to deal with. The drugs are very expensive, but yeah. And then there's also raises a lot of health equity issues too, because of the expense of the drugs. But I agree, it is something we're going to have to tackle.
Another question from Michelle.
Michelle Farrar, Paediatric Neurologist. I'm fascinated by your data, which is very compelling, but it starts at midlife.
Yes.
And so I just introduce the concept that, you know, even the tau and the biomarkers pre-symptomatically are already detectable, and perhaps the underpinnings go back further. And in paediatrics, we're seeing huge amounts of obesity and poor lifestyle. So I think, what's your comment on modifier risk factors in early life?
That's also a great question. One reason we talk about that is because we don't have much epidemiological imaging data from younger cohorts. So we know things like children who've been exposed to air pollution in Mexico City have high burden of amyloid and tau because of neuroinflammation. So it's from autopsy studies, but Some of the studies on type 2 diabetes are showing brain changes in adolescents. So you're right. And then some of the epidemiological work is now starting to look at the impact of lifestyle on cardiovascular health in adolescents and young adulthood. So we agree that there's big gaps in our data, and that is one you've identified.
Thanks. Is there any online questions at all?
No, that's me.
Oh, that's you, right? Okay. Yes, go please.
Hi, I'm Helen Goldsmith. I'm the Research Development Manager for SCHN. I had a flip side to that, and that was understanding that there's some blood tests that are able to be, undertaken, biomarkers. Is there any method in a madness? I mean, I know that, sometimes we can be overwhelmed. We feel that we may have some sort of difficulties or understanding that we may be predisposed to dementia. Understanding if there is a higher risk for myself, knowing that those biomarkers prove something, would there be, I suppose, a need to then change my lifestyle in order to address the risk factors that would increase my chance of getting it?
Absolutely. I mean, if you know early on that you have a biomarker for Alzheimer's disease, you would want to be doing absolutely everything you could in the modifiable risk factor space, as well as when effective treatments come, you'd want to take them. So, but there's a lot of issues to be worked out here about what patients are told. What we don't have enough information on the thresholds. I'm not a clinician, so. But there's a lot that's going to have to be worked out in how these are delivered, and we have to train medical staff in understanding the blood results and giving the findings to patients, et cetera. So when I've just been in a meeting in The Hague on dementia, and people are talking about needing this whole infrastructure around these new tests and these new drugs, and we don't have any of it here. We can't even process those tests in this part of Sydney. We don't have the machine. So yeah, it's a huge issue that we need to address. And given the sheer numbers of older people, just statistically, it's going to become hit us, as all of this becomes available and people start asking for them.
Very similar to the PSA debates that are happening.
It is going to be like a PSA, and that was at the recent meeting in Amsterdam. That is how it was being presented, that you'd be given there's different, it's like a cancer risk, a 0, 1, staging, and then, that would indicate different treatment pathways, etc. So that's the draft classification that's up for discussion at the moment by the American Neurological Society. Yeah. Thank you.
Thanks. I have one question. I think there's no more in the audience or online at this point. A lot of the work that myself and my team have been doing is around the cardiovascular, cardiometabolic syndrome in people living with mental illness, and that reduces, creates premature mortality, etc. And a lot of those risk factors are the same, as you know, and a lot of those genetic, interesting, the ApoE and those sorts of things. Is there work already being done trying to combine, because your field of work with prevention of dementia and the field of work trying to prevent other cardiometabolic health conditions, they've got so much in common, and really what it is from young age and included in the paediatric population. Let's do as much as we can lifestyle wise, do the biomarkers. Is there some work that's crossing over, because this is what the whole society needs?
Look, there is some. We actually have a grant at the moment to develop a risk tool that is predicting risk for cardiovascular disease, stroke, diabetes and dementia. So with 4 of the outcomes. This is another topic that comes up a lot in policy discussions, because medical research funding is siloed by disease. And then there's also a lot of stakeholders in each of these chronic disease areas, and it's hard to bring everybody together. But clearly there's also very limited public health dollars. So clearly by working together and having some unified programs that can address multiple chronic disease outcomes, it's much better for all of us. But some of the structural barriers are there, I think, that are preventing us doing that.
One more question from Toby.
Can I just ask, in terms of the concept of limited health dollars, where do you think you get more bang for your buck in terms of managing this as a population problem? Is it from prevention, active lifestyle change versus therapies?
That's a That's another really good question. I think that, well, I think the public health approach, I'm a real advocate for the public health approach to dementia risk reduction. You know, shifting the whole population level of these risk factors. If it just, because it's so highly prevalent and the risk factors are so common, those two factors combined mean that you're going to have a much better economic benefit. So there already are, so in the US, there are very high-tech brain health clinics where we could go and we could be completely worked up. You get risk modification, drugs, everything, but it's really, really expensive. And so the number of people who will, and in Europe, the brain health clinics are starting to have brain health prevention clinics where you go and get that really tailored intervention, but it's only ever going to be accessible to a small number of people. So I think from a government, you know, public health point of view, given the aging population, we need to really step up the population health approach. But that's a life course approach, and it also feeds into things like inequality and those things, yeah.
Great. Well, just in the interest of time, I think we're going to need to wrap up, but I just want everyone to say thank you very much to Professor Anstey. Yep. And we're just going to switch over the slide set, and whilst that is happening, I would very much like to welcome Professor Michelle Farrar, who is Professor of Paediatric Neurology in the School of Clinical Medicine at UNSW and a Specialist Child Neurologist at the Sydney Children's Hospital, leading the Rare Neurological Diseases Clinical and Research Program. So the other end of the spectrum, but equally important, and thank you very much for joining us today. Okay, over to you, Michelle.
Thank you, and good afternoon, everyone online and in the audience. So I think it's quite challenging to hear the words childhood dementia, and really to sort of, I think the purpose of today is to introduce the term and the rationale. And I think really, as we heard, that dementia is many diseases grouped together, even in the aging population. So although it does mostly affect old people, it is not inevitably just due to aging, is really the main message. And so today, to take you through what it is in childhood, some facts and statistics, the symptoms, the psychosocial impact, diagnosis, pathophysiology, and some initiatives that we're just starting to grow. So Dementia Australia defines dementia as a collection of symptoms that are caused by disorders of the brain. It's not one specific disease. So you can see from the definition that it could involve childhood as well. And in fact, in children, dementia results from brain damage from more than 100 genetic conditions. So you can see that the underpinnings are quite different and predominantly monogenic. But there are non-monogenic conditions like measles or Creutzfeldt-Jakob disease are some examples of that too. And so this is really how we envisage dementia. And I think it has a very distinct backdrop in that as a paediatric neurologist, we really have that trajectory of development where children are acquiring skills and growing, and their brain is also developing. And that would be denoted by the pink line here, where developmental progress is going up. And intellectual disability is where the rate of acquisition of skills is less and the gap between you and your peers broadens, but you're making progress. But in contrast to that, dementia is actually based on the regression or the loss of skills, going backwards and losing. And this graph is actually depicted based on Batten's disease or neuronal steroid lipofuscinosis, but really to say that you can be born and already have symptoms of dementia and not really have acquired many skills, but then go backwards, or it could happen in later adolescence. So the decline can be quite variable. And so similar to what Kaarin did with data and looking at global prevalence around the world, we collaborated with the Childhood Dementia Initiative in around Australia to actually undertake a scoping review of all genetic conditions that would fit under the umbrella term of dementia in childhood. And in fact, there's 170 conditions, less than 5% have effective treatments. And we were able to find approximately 70 conditions where there were actually data on incidence, prevalence, age of diagnosis and survival. And we analysed those, the ones that were untreatable. And when we took those all together, the incidence of those 70 was 1 in 2,900 births. That's the same as cystic fibrosis. Why don't you know about it? Because these kids don't survive. And so when you look at that around Australia, what that means is there's 107 children or babies being born every year with a dementia condition. And when we looked at the survival of it, there's 90 premature deaths every year. That's actually, when the childhood cancer statistics are, there's 89 deaths from childhood cancer per year. So it's a pretty big problem. And the cost is enormous, both economically. I think it's $395 million a year. It might be, I'll have to check that. But I think you can't quantify the cost to the families and to society that is lifelong for those carers. It's immeasurable. And so we looked at the data and we looked at the Kaplan-Meier curve for the conditions where there were data. And there's some conditions that are so rare, there's no data. And the median survival is the age of 9 years for a child. About a third reach adulthood and 10% over 50. But really the take-home message is that there is a wide spectrum. Some children do die in infancy, like Gaucher's disease. There's other children that do make it to 50, like Rett syndrome. So from the survival analysis, you can see that the statistics do show that this is an important problem. But I think it's really the challenge of rare diseases where they're individually rare, but they're collectively common. And so the rationale is that by bringing these together and taking a collective approach and looking at what's common, hopefully we can make progress and amplify it and accelerate it. And so when we look at the funding and resourcing and the attention that childhood dementia currently receives in Australia, it's actually fourfold less than most other conditions. And I'm not trying to take away from those at all, but just to say that there's huge unmet need. One example is clinical trials for new therapies, 12-fold less than trials in childhood cancer. So really we would like to be able to mount the case to be able to attract really leading clinical trials to really make a difference in this population. So let me introduce you to Angelina, and I got this off YouTube, but it runs for a while, but it really tells quite a compelling story. I hope so. very hard to watch. So I think you can see from that video that the symptoms are quite similar to what adults with dementia get: a decline in cognitive ability, problems with attention, concentration, memory loss, learning difficulties, problems with thinking, reasoning, confusion, disorientation, behaviour changes, emotional changes, personality changes, sleep changes, loss of speech, incontinence. So you can see there's many similarities across the life course. So Jason Jafar, who was an honours student who worked with our team, collected or found 45 children across Sydney Children's Hospital network with various dementia syndromes and confirmed diagnoses in 4 months. And he did a survey and looked at what the symptoms were. And you can see here that the most, some of them were pre-symptomatic, which is why they don't all have cognitive impairments, but you can see the spectrum of symptoms and how common and how prevalent they were. And this really highlights the challenges that the families are facing day to day and the real need for care and support for these families. And the colour on the side is the Strengths and Difficulties questionnaire, which is a behavioural assessment we use for children. And it really shows the proportion of children in this cohort who had an abnormal result. An abnormal result is above the 90th centile compared to their peers. And you can see here that 35% in the red are scoring abnormal scores for total behavioural activity. So it's threefold higher, more than threefold higher than their peers. And a 60% scoring high for impact. So that's the effect of the child's behaviour on their home life, their relationships and their learning. And so in the subscales, it was hyperactivity and social behaviour. So really shows the need again for better care and support for them. So the mean age of diagnosis was about 2.5 years. This is from our literature review again, but the mean age of diagnosis was 4 years. So it really shows the odyssey for, I think most people would face with dementia to get a diagnosis and the need for pathways for clear diagnoses. So I guess in terms of prevention, this is very exciting. It came out yesterday. This is a case study of X-linked adrenoleukodystrophy. So adrenoleukodystrophy is an X-linked condition. You might have watched the movie Lorenzo's Oil, where the child was affected with adrenoleukodystrophy. And it can affect the adrenal glands with their Addisonian crises. It can cause a dementia, it can cause a myeloneuropathy. So there's various different manifestations, but typically boys would develop symptoms in late childhood, but by the time they're diagnosed, you can't treat them. But there is an effective treatment if it's picked up early. It's a bone marrow transplant. So really, based on my work in spinal muscular atrophy and my experience with newborn screening and health policy to get that onto newborn screening, I partnered with the Leukodystrophy Association and the Department of Health to request the addition of X-linked adrenoleukodystrophy to the newborn blood spot screening, and that went through the MSAC pathway over the last 12 months. And yesterday it was announced that it's been recommended by the Health Minister for the addition to newborn screening. And so there's a lot of work to do yet to work out what the best path for health implementation is, because it's going to be challenging. But really, I think the partnership, in the words of the parent that I partnered with, was, I think you can still provide care after a child passes and healing. And really the quote of this father, who is now quite elderly himself, who promised his son on his deathbed he would do something. And he's been trying for many years, but he says it will save the worst pain that exists on this planet. And he said, I'm going to sit quietly and absorb this today, and it's healing me and giving me passion, hope and excitement. And really, when you think that there's 170 genetic conditions that cause dementia, there's a lot more work to do. So I am very privileged to be part of a project looking at new technologies for early diagnosis in newborn screening with genomic technologies. We aren't saying that we should be doing them, we're exploring it, and it's a very open question. But can genome sequencing have a role in newborn screening? Can it be done at scale? Can it be done at speed to be able to diagnose children early? Because 50% of boys with adrenoleukodystrophy in this country are diagnosed and you can't offer them a treatment because it's too late. It's only when you get a younger sibling and you test them and you know that both boys are affected, you can treat the younger one, but not the older one. Is it acceptable to do this? Can it be used for other purposes? And how can we store the data and use it in a way that maintains trust and privacy and security in the population? So there's the possibility, and we're really exploring it. We're not proposing to implement it at the moment. So really, I think taking it back to the families, it's devastating, not just for the child, but also for the family. And Claire Wakefield and Suzanne Nevin have done a lot of work on this in the Behavioural Sciences unit. And really they undertook a scoping review of psychosocial experiences of caring for children with dementia, and they put together the outcomes of 19 articles, and it was a perspective of 1,838 parents across 25 different countries, different dementia-causing conditions. And there were three main themes, and really the parents highlighted the extensive difficulties connecting with medical teams and navigating their child's rare life-limiting condition that was progressive. And there were many, many psychosocial challenges. They were physical, social, and economic, emotional, and psychosocial. And really, I think it really sent a message that there needed to be access to coordinated care, community-based psychosocial supports. And really, the other thing that came out, that when these were in place, parents coped better, they had more resilience and less isolation. So I really hope that connecting across the life course, we can learn from our colleagues in adult dementia and really take these lessons into the paediatric sphere to improve our care. So I think there was a need to prioritise integrated care with families, and the analysis showed the need to prioritise this, but also tailor it to the distinct stages across the child's trajectory. Families report that care and support is inadequate, it's poorly coordinated, it's fragmented, it's inconsistent. And I think the outcome of this has been a commitment from Dementia Australia to expand support services to children living with dementia. But it also highlights the need for more research to really understand what the systemic improvements can be. And to really, I think another lesson from my colleagues is that if we can show that multidisciplinary clinics improve outcomes and quality of life, and hopefully we can scale them, because at the moment there is no multidisciplinary clinic for these children. And I think there's the potential for cross-pollination and collaboration in child and adult dementia research and to improve clinical systems. What we've also done, and this was again Jason, the honours student's work, was he interviewed 26 of the families around the term childhood dementia, because it has to be a term that's acceptable, and I find it challenging even now. And in fact, 73% of them were supportive. And the reasons were they thought that a collective approach, not what's your disease, it's so rare, I've never heard of it, it's not relevant, I'll never see it again, is that it empowers better advocacy, awareness and communication. 23% of parents agreed with the benefits, but they thought that uncomfortable with using it to describe their child. And I get that. They're children. They're not people with dementia. They're still children. They are growing up. They still like to play. They still like to dress up. And they want to be treated as an individual, not a number, and also a child. So really the take-home message is that there's a lot, it's a nuanced term, it has to be used carefully, but there's a lot of education around the use of the term to build awareness and acceptance of that as well. So I think further caregiver perspectives from these interviews were that there's definitely opportunities as well as barriers to really improve management, that there are currently gaps, but gaps are always opportunities. So the parents are saying that they feel like they need to fight every day for their child to get the care they need. It's hard to find services for children with complex communication needs. So this graph really shows up the top, it's the brain health in the domains defined by the World Health Organization: cognitive, sensory, social, motor, and emotional. But below it, you can see the diagnostic categories. So childhood dementias can be lysosomal storage disorders, mitochondrial, peroxisomal, other inborn errors of metabolism, and a very big group of other disorders. And each one of these from Jason's study showed that they affected every domain of brain health. But then when you look below the umbrella, each one of those also had common disease mechanisms and overlaps and pathways from a pathophysiological level. And they're actually quite similar to the pathophysiology of adult dementia syndromes: accumulation of macromolecules, there's accumulation of tau. There's also neuroinflammation, sometimes as a secondary response. There's also energy failure and organelle dysfunction. So some of the other work we're doing in our collective approach is we're also looking for biomarkers, because we understand the challenges of developing therapies, but with so many genetic underpinnings, I think with advanced therapies, gene therapies, ASOs, RNA therapies, there's a real potential to amplify the impact of these, but we can't take clinical trials in N of ones. We need to know how to measure outcomes, and biomarkers of common pathways as surrogate endpoints that ultimately translate to clinical impact are a way to sort of potentially accelerate the pathway, but also look at those that can be targeted collectively. So we're currently undertaking a biomarker study to identify biomarkers, discover them, and potentially help us in the future with clinical trial design and therapeutic development. And that's an initiative where we're collaborating across Sydney Children's Hospital Network with Kids Neuroscience at Westmead and Randwick, hoping to get 70 children's samples to look at this collectively through an MRFF grant. So in conclusion, I think while dementia is typically assumed to be a condition of aging adults, children are also affected. It's important that we take a collective approach. It is a phenotypic syndrome as well as individual. And I think this is the approach with adult dementia, and we'd like to take that approach as well. And we believe that the commonality of the presentation impact can build powerful collaborative opportunities in research, care, support, advocacy and awareness to create change. Thank you. And I'll just acknowledge, so I think what we're doing, that was just, I'll skip over that, and just lots of people to acknowledge across Sydney and beyond. Thank you.
So much, Michelle. Wow, that's really eye-opening for many of us. And your work has been, the work in this area of rare diseases has been really something to behold in terms of advocacy and modelling clinical care, as well as changing practice and policy. Very inspiring. So thank you. Are there any questions from the audience or Sam online? Toby, go ahead.
So these are collectively common but individually rare, and so I suppose that is going to be a real challenge in terms of if you want to try and actually develop active therapies. I mean, lucky that a couple of the storage disorders you can transplant. It's not necessarily a great therapy, but you can do it. But I suppose, how do you see driving forward to try and actually get specific therapies for some of these quite individually rare diseases? Because that seems to me to be one of the real tough challenges.
Yes, but I'm also surprised because there's a lot of rare cancers that have targeted therapies. So we take our hope from you. I do think you can treat upstream and downstream. So we know what the genes are, and we can take a technological platform approach. You can have an AAV vector and deliver the gene if it's amenable to that. But I also think there's downstream approaches where if you can ameliorate the inflammatory response and give the cells more energy, it's going to be a cocktail of therapies, much like chemotherapy.
Is there any other? Oh, there's something else in the chat. Oh, that's you. Oh, okay. No, that's great. I think given the time and really what we've just learned and also the video we've just seen, I think absorbing this from everybody is it's going to take some time, but no doubt people, if they're interested, will reach out to yourself, Michelle, and also to yourself, Kaarin. That's been an incredible session, and really shows the power of working together across disciplines, across clinical, translational settings, biomarker settings, genetic settings, and really very grateful to all of you for the work that you've done, and thank you to the precinct for bringing us together. Let's keep doing these presentations, but also the work to make a difference in people's lives. Thank you very much.